Carnegie Mellon is a great place to study computational biology because Comp Bio really fits with the message and the goals that CMU has. CMU has always promoted interdisciplinary research and finding ways to create novel research between very different departments. Comp Bio requires work and collaboration between mathematicians and physicists and biologists and chemists and computer scientists. And so it's really in keeping with what Carnegie Mellon is promoting in its students and its professors.
CMU is a fantastic place to study cell biology because of the diversity of work in cell biology that's being done. I am a member of a membrane trafficking lab, and the other two labs are Adam Winstead and Tina Lee. We actually have weekly lab meetings. That's a joint lab meeting between all those three labs.
Carnegie Mellon is a great place to study developmental biology because there are several developmental labs that we can share resources with, bounce ideas off of, and even frequently meet with.
I work in Nathan Urban's lab, and his work is all centered around the olfactory system, so the sense of smell. And specifically, we work in rodents, we work in mice. So we have an animal facility that houses mice, and we're able to, for me specifically, we're actually able to take mice and actually make very thin sections of their brain and actually record from neurons in the olfactory bulb and try to, from the data that we gain from recording these neurons under different conditions, try to understand how is the olfactory system itself processing information. And the overall goal of this is just to understand more about how circuits in our brain process incoming sensory information that we receive all the time.
So our lab focuses on the molecular mechanisms of ribosome assembly, specifically the large subunit. So ribosomes are such complex structures which are integral parts of the central dogma of molecular biology. That's why it's really an interesting field to delve into.
The main goal of the Hemi Lab is basically to elucidate these gene regulatory networks in echinoderms, and so sea urchins and starfish.
In the Macbeth Lab, we study ADARs, which are proteins that interact with nucleic acids, specifically RNA, possibly DNA. That's one project that we're working on right now. And here is human ADAR2. This is just the catalytic domain, so we've had truncations made of the protein. And this is the actual crystal structure.
So the Campbell Lab is really interested in studying the formation of multi-tissue units. By multi-tissue units, I mean the formation of muscle side by side with tendon, side by side with bone. And so in a dish, you should be able to create 3 separate populations of cells: muscle, tendon, and bone. And ideally, they'll be, purely muscle and purely tendon and purely bone. So there's the issue of really controlling the heterogeneity you get when you put all your different growth factors and cues for differentiation in the same dish. And with the inkjet printer that Phil Campbell and Lee Weiss, designed together, you're really able to take advantage of this inkjet printing technology to create these different patterns in vitro in a dish, which you can then transfer in vivo into mice.
The goal of this lab is what some scientists call molecular, non-invasive molecular imaging. It's a relatively new field, and I would like to think that we are at the forefront of it. The idea is to be able to image molecular and cellular events that are happening in a live animal, and that would give you a unique perspective on how behavior and various diseases happen on cellular molecular level without actually chopping up pieces of tissue or growing single cells in the laboratory. So the goal of molecular imaging is to be able to image non-invasively events that are happening inside an animal and sort of predict early development of disease or correlate sites of behavior with true molecular events that are happening on a micro scale.
The Schwartz Lab has 3 main projects. We have a phylogenetics project that's looking at classifying families of different gene families over time. We have the cancer project, which I rotated on in my first rotation, and that's to understand the evolution of tumor cells inside cancer patients. And lastly, my project in the Schwartz lab is on viral capsid assembly and trying to understand how viruses assemble in vitro, starting from individual protein components until they form an entire capsid.
The theme of my project and the theme of most of the projects in the Lee Lab actually is to understand the structural components that cause the endoplasmic reticulum to look the way that it looks, to have the morphology that it has. In particular, I'm working on a protein called elastin, and it turns out that this protein, when you knock it down using siRNA, causes the ER to change its morphology. And so what I'm doing is a series of sort of rescue experiments to start to find out particularly what it is about the elastin protein that structures the ER in a normal circumstances, and what we can do to change how that protein structures the ER. And that is really interesting, I think, from a broad perspective, because there aren't very many things that have been identified that are required for ER morphology. And indeed, the mechanisms for how the ER morphology is generated and maintained are also not very well known specifically. So what I want to know is what is elastin doing? Is it using ER membranes or in some way structuring three-way junctions? It's hard to know. And so as we start characterizing specifically what the phenotype is, we can start to understand a novel ER morphogenesis mechanism.
The project that I'm working on is crystallizing human ADAR2 with its substrate, and we're trying to better understand how protein nucleic acid interactions occur.
For my project in the Minden lab, I'm studying ventral furrow formation and its cytoskeletal regulators. To do that, I've designed a technique called end-on imaging, where we orient the embryos, the Drosophila embryos, in a unique way so that we can image through their posterior end and simultaneously image the cell shape changes along with the proteins that regulate those changes.
So my particular research is in studying the molecular evolution of multi-domain proteins and trying to figure out the origin of varying domains and whether proteins that have the same domain content actually evolved from a common ancestor, or if they had evolved that content independent of one another, so it evolved multiple times.
I work in the field of cancer phylogenetics. We use whole genome data like microarray gene expression data or copy number variation data and apply novel approaches to extract meaningful information, which can then be used to build phylogenetic trees which help us determine the sequence of mutations or abnormalities that define the growth and development of cancers. I work in breast cancer in particular, and the goal is at some point to determine significant biomarkers and progression pathways which can contribute towards novel therapeutics, especially targeted therapeutics and personal genomics and personal medicine.
A project that had been going on in the lab is to determine how sea urchins can make a normal skeleton, and I'm trying to figure out the molecular basis for these changes.
My job is, as I mentioned earlier, is with inflammation, and inflammation really sets the stage. It affects how successful this differentiation is going to be. And so we're really interested in controlling the inflammatory process, we're interested in controlling the differentiation process, and we're also interested in being able to verify and quantify how successful your differentiation was.
The summary of my dissertation work essentially is moving the study of ocular dominance from a classic systems neuroscience perspective into proteomics. So what I do is I take samples, brain samples from functionally defined ocular dominance columns, which we determine where these columns are in an in vivo imaging setting. And I take biopsy samples from those columns and I move those through a proteomics pathway to try to characterize the protein differences between a left eye and a right eye ocular dominance column.
This being an adrenergic receptor, it responds to epinephrine, which is adrenaline. It's very essential for a lot of your body's signaling and responses to stress and sudden events, allowing you to respond quickly, and has even been implicated in heart problems if you have failures in the signaling pathway. So not only is it a very interesting model receptor for understanding how this whole group of receptors works, but as an individual receptor, it's very interesting.
Well, the advantages and the implications of the research that I'm doing in the Schwartz lab is not only to understand how viruses assemble inside cells, which have wide-ranging applications, and just from basically it's the knowledge base that you gain from understanding how viruses work, to being able to, if we understand how viruses assemble, we have a better idea of understanding how to prevent viruses from assembling. And that's with different therapies and drugs that could be produced in the future.
In the big picture, research such as this with phylogenetics is very helpful in medicine, in designing experiments in model organisms, where you can identify which genes or proteins are orthologous to the ones in human that you might be interested in.
Our hope is that our findings can provide useful information which experimentalists can then use to guide their experiments, which would then be crucial, and the crucial link between translating the research into everyday use.
It could also generate a way for us to understand the disease phenotype in humans. So a mutation in elastin in humans causes retrograde spastic paraplegia, so you slowly lose the use of your lower limbs. And this is not a very common disease, but we don't know what's going on and what's causing that. And so if it turns out that a perturbation of ER morphology is what's causing that, then maybe we can start to address a solution to that. So that would be neat.